Hello, and welcome to Beyond Biotech, the weekly podcast from Labiotech. My name is Dylan Kassane, and this is episode 197 for the podcast. Today, we're digging into a topic that's getting a lot more attention lately: how early decisions in cell therapy end up shaping or complicating everything that comes later. Our episode today is Freeze Variability, not Progress: how to strengthen your cell therapy supply chain from the start. And we're going to challenge a few long-held beliefs about how starting material should be handled. My guest today is Dominic Clarke, Vice President of Technical Operations for IntegriCell at Cryoport Systems. Dominic has spent years in the trenches building and scaling cell therapy processes from early development through commercialization. So he's seen where things break and what actually works. I hope you enjoy my conversation with Dominic Clarke. Dominic, it's great to have you here today. We know Cryoport Systems well here at Beyond Biotech, but for anyone tuning in who might be new, can you give us a quick refresher on what Cryoport Systems focuses on, and maybe tell us a little bit about IntegriCell and how it fits into the bigger picture?
Yeah, absolutely. And first of all, thank you for having me here today as we dive deeper into the industry norms and challenging some of those norms for the cell therapy supply chain. As you said, Cryoport Systems is a global leader in temperature-controlled supply chain solutions for the advanced therapies. We support the full vein-to-vein journey to protect irreplaceable materials, but it's because of that end-to-end visibility that we see firsthand where variability and risk actually commonly enter the system. And it's this unique position that allows us to translate those direct insights into our solutions and best partner with our customers to enable patient access at a global scale. IntegriCell is a part of that complete platform that focuses specifically on the incoming patient starting material. IntegriCell essentially enables consistent cryopreservation of leukapheresis starting material through a standardized automated platform or process that we've developed and put in place. And our goal within IntegriCell and across all of Cryoport Systems is to simply build a reliable foundation that supports the development, the scale up, and certainly the most important aspect is long-term success for our customers.
And before we get into the technical side of things, I'd love to give listeners a sense of what you personally focus on. Where do you spend most of your time at IntegriCell?
Maybe I'll just kind of step back for a second and maybe it'll help to understand. And so I've been living and breathing cell and gene therapies my entire working career. And, I would say that, I don't know if this is going to make too much sense, but I kind of look at myself as a translational business entrepreneur with a scientific foundation, right? So my background is all in, essentially started with low temperature biology and cryopreservation, and that's where I essentially got thrown into this cell and gene therapy space, working to develop a cryopreservation media, and not so much to develop the media, but to establish a norm in an industry when it was in its infancy. So, and you'll see a trend of this. At that time, the industry was essentially using processes that were academic and not thinking about a scale-up manufacturing type industrialized process. So we had to help change the mindset. This was 20 plus years ago, to get the industry to start thinking with an industrial mindset and a translational mindset and not just an academic at the bench. So we had to help force the idea to change from using and making up your own medias with the variability that comes with that to using something that's been developed and under a GMP approach with basically a standardized component that now you can put in place. And so from there was a launch pad to me getting into a number of different disciplines from the side of a service provider or a product provider. That led to working with a plastics company to develop and offer improved solutions for, again, targeting the primary container, the bag that is used to be frozen for long-term storage and to move capture the cells that we're freezing today. And then subsequently, working with starting material providers and optimizing cryopreservation approaches for healthy starting materials that people were using and companies were using in their development and process development, and scaling up to manufacturing, and then ultimately now working here at Cryoport and IntegriCell. And then so you're back to your main point of your question. What do I do every day? So I oversee the technical operations for IntegriCell, and that includes the process development, the MSAT or tech transfer teams, as well as the operational teams that are doing the cryopreservation and performing that service for our clients and the patient material. And those groups coupled together is really where I can combine all the feedback from the industry and the feedback from the clients. And we're not just building and offering a standardized solution today. We're constantly looking at how do we continue to optimize that service and that cryopreservation offering so that we can continue to advance the field and support the needs of the industry.
Look, let's jump into the heart of today's conversation. In cell therapy, we talk endlessly about innovation, yet a lot of workflows still rely on assumptions that were created a decade ago. One of the biggest is the idea that fresh cells are always better. But is that actually backed up by data, or is it just a habit we've fallen into?
We get used to the norm, right? Fresh workflows are often, they're considered, it's considered normal. So they're often inherited from an early academic process, for example. Oftentimes they're simply, that's what we're used to doing. And so whether it's an early academic process or even a little bit more established biotech, this is the workflow that the community is used to. And so why change? Ultimately, there's a lot of challenges that are associated with fresh that many of the audience are familiar with. And, the fresh is just accepted, and it's inevitable with this variability that comes with it, the scheduling risks, and ultimately the scalability is just, again, perceived as the norm. So operationally, there's a better way, and variability doesn't have to be inevitable. So standardization is a strategy that the entire industry is looking at. But, we think about standardization as a strategy, and cryopreservation is certainly the mechanism to achieve that kind of standardized approach to reduce the overall variability, specifically at the front end where that variability is super high when you're talking about patient material that has its own inherent variability, and then the process that has to go through. So by offering a standardized approach with a cryopreservation, that's how ultimately what we looked at as the launch pad for IntegriCell.
And even when teams do introduce cryopreservation, it's usually late. It's sometimes during scale-up or even during tech transfer. Why does it tend to happen that way?
Yeah, I mean, it's again back to the fundamentals of these inherited processes and what is considered normal or just standard practice, right? It's why change, even though we all know that there are inherited risks and challenges with it. It's easier sometimes to just go with what they're used to. So a lot of the early programs are designed around immediate impact, and that's what works fastest in the lab and in the clinic. Again, if you think about a lot of these companies, that they're trying to get to the clinic, there's a short time period, and the runway is quick, right? So fast, you want to get to the clinic quickly, and sometimes adding in a change to a process takes time. It may be seen as something that is useful, but they don't have the resources. They don't want to take the time. They just want to get to the clinic, and they'll deal with it later. That's the typical way that a lot of companies look at this. And then you think about, again, to that idea, fresh workflows, they feel simple at first. It makes sense. We can do this small scale. We can control a couple of patients. We can move the product around. Oftentimes, again, the clinic that they're in, the patient material are right next to where they're going to be doing their manufacturing. So they can manage the logistics, get there as fast as we can. But, those early decisions become locked in at that point. Once you get into the clinic, it becomes a lot harder later on down the road, and certainly more risky to make that change. And so what we found, what works in the lab and then in the clinic at these early stages, just simply doesn't scale very well ultimately. And so programs are then forced to have to pivot when it's late in the product life cycle and the regulatory scrutiny is significantly higher. And then at this point now, these pivots become super costly and time consuming. And so it's really trying to change the mindset to, establish and make these changes earlier on.
Yeah, right. And I guess it leads to just that really obvious question. If you're adding freezing late, you talk about these risks. What actually happens? What sort of challenges show up at that point?
Complete overhaul in a sense. You're asking regulators to accept a major process change, and operationally now you're introducing some uncertainty. You're adding in new points of potential failure, new challenges, new validation requirements, and new training needs, right? So from a sponsor standpoint, you now have to consider, the starting material characteristics, the variability and the reproducibility. You got to deal with ultra low temperature storage, cryogenic handling considerations, and exposure to aspects like mechanical and thermal stresses. So yeah, these are new complexities that are introduced, but the risk mitigation benefits, as we've seen over and over again in the industry, far outweigh any of these additional considerations. And you allow yourself ultimately much better planning, manufacturing scalability, and the ability to plan for global growth. And that's just something you can't do. It's difficult to do with a fresh workflow. So, and then, if you build in cryopreservation and it's built into the SOPs and your standard operating procedures from the start, you don't have those disruptions, right? We talked about it earlier. Yeah, you have to change your mindset and you have to add some time and cost up front if you think about cryopreservation at the beginning. If you have to do it later on, these becomes so much more challenging. And so if you build it in from the earlier stages, freezing becomes a proactive design choice and not a downstream reactive fix.
Dominic, you mentioned variability, and let's talk about that for a second, because it's a word that everyone uses, but not everybody really specifically defines it. From your perspective, where does variability actually hit hardest in that cell therapy workflow?
Variability issues surface probably from two major avenues, right? Time and people. One of the bigger ones that are most common to everybody is that you have different operators, right? So you also have variation from site to site. You have scheduling discrepancies. So cryopreservation allows you to separate the biological variability from the operational variability. Now, because this variability enters through things like manual handling, open processing steps, you have different operators, so you have to make on the cusp operator decisions. You have scheduling pressures. We try to take a lot of that type of variability out of the process, right? And so we adopted and developed an automated closed process, or our ACP, within IntegriCell. And the goal was to create a repeatable, standardized process that reduces as much as possible all of those time and people variations. And much like we were talking about before, the value of the ACP process is highest when it's embedded at the earliest time frames. And then so again, your SOPs, your operating procedures are built around this automation scale, and more smoothly allowing you to reduce the need for these late stage process changes. And that end result is continuity, right? From early development all the way through to a commercialized process.
You mentioned something there, that automated closed process, the ACP. Can you walk me through what that actually looks like at a high level?
Yeah, at a high level. So essentially, if you receive in a patient starting material, essentially you receive that, and the next step is to wash and concentrate that incoming blood-based product And then to do that, there's if you're doing a manual process, this is done with a centrifugation step. And then you do the manipulation. And essentially you get a washed concentrated product that is now formulated with the cryopreservation medium and subsequently frozen using a controlled rate freezer. The automated process takes a number of those handling steps where you have to handle the incoming material and add in your buffer to wash it. Then you have to remove that. Then you have to, as you concentrate it, these are all manual handling steps. Again, this is something that you can do, but ultimately we went ahead and built this automation in using a system from Fresenius Kabi called the Q, which takes all of the manual manipulations out of it. You attach the incoming blood product to the system. You've got your buffer material already there as well. Essentially hit the start of the process, and the machine takes care of most of it itself with some steps in there to just, evaluate in process cell counts and so forth. But then it's, also at the same time, at the end of that wash and concentrate cycle, the cryopreservation medium is then added to that product. So your formulation is performed on the automation, and it fills the final product bags at the same time. So taking the manual manipulation out of it. So I think that generally is the background of the automated process that we've developed.
Yeah, I think that makes sense. But what if someone's already got an existing process? Is IntegriCell still a fit for them?
Yeah, I mean, I think it's a great point. We, as I mentioned, as I also, oversee and manage the process development and MSAT team. So we are fully capable to support if a client comes in with their own cryopreservation process. That's great, right? They've been proactive and thought about adding cryopreservation from the start. So we have a full team to, complete the tech transfer process, so we can, essentially bring on board their process and run that process at our site. And I think another important piece to ensure that, we cover is premise of IntegriCell isn't just based on, having an automated standardized cryopreservation process that we've developed. It also extends beyond the idea to offer this standardized process at multiple locations, both regionally and globally. And that's a big piece, right? Because whether someone comes in with a manual process, one of the big challenges is having a manual process or any cryopreservation process today that's offered by a different partner, right? So if you're having an incoming patient material at a hospital system, and they can cryopreserve right there. So they're going to use a process that they developed, or maybe it's the client developed process, but then you have 10 or 15 more unique sites offering this, a similar type of process. They have their own cryopreservation process, similar because it's manual, but it's slightly different, right? And so everybody's doing something different today. So again, we recognized that that's a gap. And so not to just build a standardized process, but to have multiple locations so that you can offer that standardized process. And that's the same thing we can do if we bring on somebody else's process. Even if it's not automated, even if it is, we can offer and deploy that same process at each of our locations, which improves reduces the variability from process to process, from site to site, but it also offers a standardized quality system across the board as well. So in a longer description than you probably intended, I wanted to make sure that we can cover process development, optimization for groups. We can provide the tech transfer of their own process, and we can also offer our standard automated process, which again covers the globe.
Yeah, no, we don't mind the long answers, and it really helps to paint the picture. But I guess despite all the control that freezing and closed processing brings, a lot of teams are still going to feel safer with fresh cells. Why do you think that belief just persists?
Historically, freezing did have its own limitations, but I think we've certainly seen that the science has moved move beyond that. And in practice, I'm not going to discount the fact that fresh is a viable option for some, and some of the commercial therapies use an incoming fresh material. So I don't want to discount that that's not, again, if you could do it, fresh is a very, is a great way to go if you can control and you have the resources, and very few companies have the resources to be able to, use a fresh incoming material. And then, as we know, in practice, fresh introduces so much more risk because it just simply removes the flexibility. You have a short time period to move that material around from the time it's collected to the time you have to manufacture it, without every hour, every day that passes while it's being transported and perhaps being held up means you're losing viability, you're impacting the quality, and every product's different. So you have inherent variability that we talked about before. So, if you take all that aside, everything improves operationally when it's frozen. Manufacturing then becomes decoupled from the clinical timelines. The inventory becomes manageable. And most importantly, the patient is dosed on a time, based on, dosed on time and reliably. So I guess if I, really had to think about summing this up, fresh optimizes for immediacy, and frozen optimizes for scale. And again, fresh works really well when you're at small scale and you're close by. You can control that, but as soon as you have to scale this up, fresh is not your friend anymore, and cryopreservation and frozen becomes your new friend for sure. So I think today we're still trying to change that norm, and fresh is the answer, for many today, but it's certainly not sustainable, and it's not forward thinking. It's not repeatable. It's not the reliable process flow that you really want in your pivotal trials moving into commercialization.
And of course, that big concern, the one that comes up, is always quality, viability, potency, overall cell performance. What does the data actually show?
Well, data, shows that when cryopreservation is properly developed and validated, we consistently see reduced variability without the compromise of the critical quality attributes. And the key is, again, a well-qualified process with optimized cryoprotectants and the use of controlled rate freezing, along with standardized thawing protocols. Uncontrolled processes, like anything, they just simply introduce far more variability than a tightly controlled freezing process ever will. From a regulatory standpoint, validated cryopreservation really strengthens your story because it improves that reproducibility and demonstrates your commitment to reducing any variability in your process from the start. When we, compared our automated closed process, our ACP, to fresh handling, the data from that study showed that the ACP preserved cell viability and recovery, these critical attributes at levels comparable to a fresh material, while ultimately reducing the operator-dependent variability. Upon further analysis, it proved again that the preserved cellular identity and the functional readiness. Phenotypic analysis demonstrated that the ACP preserves the key T cell subsets and activation profiles for functional equivalence to the fresh starting material. And this is, a study that we did in collaboration with a company called Celiad at the time. Again, all of this data demonstrated the key attributes that the cryopreservation itself over fresh and a standardized automated process can really do to improve over the standards of today, which is that fresh incoming material.
Dominic, let's zoom out a little bit. Many programs still treat cryopreservation as just a handoff, like it's a different chapter in the process instead of part of a core workflow. What does that create in practice, and how does it impact that wider supply chain?
Ultimately, handoffs introduce unnecessary risk, and more importantly, you get fragmentation with different standard operating procedures and different assumptions. When when you're in development, cryopreservation and scale up are integrated through a single partner platform strategy like IntegriCell. The accountability and the continuity simply improve dramatically. And so when each step in a process is handled by a different vendor, each handoff with those vendors introduces variation. So across cryopreservation, biostorage and transport, these issues just simply get compounded. And this is where an integrated model eliminates the variation so that material moves in a single aligned system. For us, operationally, it's a partnership and a partner who thinks end to end and truly understands how early decisions impact, their commercial manufacturing.
Before we wrap up, I want to take a look further ahead. What's next for IntegriCell and for Cryoport Systems? What should listeners expect to hear about in the year ahead?
Yeah, it's a great question. There's always a lot happening, and we're always again learning and working closely with our industry partners. So looking ahead, we're continuing to evolve our global supply chain center network to focus on the expanding global reach while deepening the local capabilities. So last year we opened a new global supply chain center in Paris, France. We'll be opening another global supply chain center in Southern California later this year. And these global supply chain centers bring all of our services under one roof to bring critical services closer to where therapies are manufactured, stored, and delivered. So this means cryogenic storage, cryopreservation support, the packaging and distribution services are all housed together to reduce handoffs and limit the risk. And, again, limit the variability that leads to that risk. So, but the value of these global supply chain centers is integration, and they're designed as connectors in a unified platform with process standards that are aligned globally for consistency across regions. Much like, I described the background and the benefit of the IntegriCell platform is to offer that standardized cryopreservation process across all sites. And so broadly speaking, this positions programs to move from the earliest stages to multi-site trials to commercial scale through an infrastructure that can support higher volumes, more complex routes, and regional regulatory differentiation. And once again, it's really about enabling scale without increasing the variability.
Finally, Dominic, there are teams listening today who are planning the next phase of their development. What's a thought you can leave them with as they're listening today?
Yeah, I guess there's definitely a lot of change, and the market's evolving and the industry norms are shifting. So I would stress, much like we talked about in the beginning, freezing doesn't slow progress. Uncontrolled variability does. So defaulting to control, consistency, scalability means defaulting to frozen and moving away from that fresh mindset. So for cell therapies, to truly scale, standardization must be treated, really as a strategic advantage. And as these are the consideration. This is the consideration, perhaps, that I would challenge your listeners to consider and simply think about it at a much earlier stage than maybe they had previously, and really think about incorporating, the idea of a cryopreserved starting material. I guess one other piece I would add to that is, thinking that mindset. Think about if you're going forward with fresh, we know the benefits of adding a frozen cryopreserved starting material. So, take that leap, right? Believe in the fact that your process and your therapy is actually going to be successful. Put the work in up front, add in a cryopreserved option, and therefore you give yourself the flexibility. If you want to stick with fresh, have that cryopreserved product ready to go, and then you're prepared for anything as you move forward. But again, think proactively and think with an open mind.
Dominic, thanks for joining us today on Beyond Biotech.
Yeah, much appreciated. Thank you.
That's it for this episode. Don't forget to check out all the latest news and analysis at Labiotech.eu. I hope wherever you are in the world, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.